Here, we're given a DNA sequence where the coding and template strands are unidentified. All we know is that this DNA codes for 4 amino acids. To find the correct orientation of the sequence, we need to identify all of the possible nucleotide sequences that code for the start codon, AUG. On DNA, it will be ATG. Then we will need to identify the stop codons, which will either be TAA, TGA, or TAG. This codon will not result in an amino acid, so it will be excluded from the 4. As we see, the bottom strand has 4 codons which code for amino acids, so it must be the correct sequence. So therefore, the bottom strand is the coding strand, and the correct orientation will be from right to left. So here we have our rearranged DNA to its correct orientation. The top strand is the coding strand, and the bottom, the template. The template strand will be read to create this mRNA, which will code for the amino acids methionine, valine, serine, and lysine. Once again, the stop codon does not code for any amino acids. Now, what would happen if one nucleotide were to be replaced by another nucleotide? How will this impact the polypeptide end product? The first type of point mutation is a missense, or when the mutated codon codes for a different amino acid from its original codon. On our third codon here, if the last base cytosine were replaced with an adenine, what would have normally been a serine will have become arginine, changing the polypeptide outcome. This change is called a transversion, a change from a pyrimidine base to a purine base, or a purine base to a pyrimidine base. The second type of point mutation is a nonsense, or when a codon coding for an amino acid becomes a stop codon. On our fourth codon, the first base undergoes a transversion change from an adenine to a thymine, which would result in a stop codon on the mRNA. This will result in a premature stop to translation, resulting in only 3 amino acids. The third type is a silent point mutation. This is where a change in the base does not result in a different amino acid. Here, our second codon undergoes a change on the third cytosine to a thymine. This is a transitional change, where a pyrimidine base changes to another pyrimidine base. The same applies to purines. This change results in the same amino acid as the wild-type DNA. The last point mutation is a read-through, where the stop codon mutates into an amino acid coding codon. Here, the last adenine of the stop codon mutates into a thymine, which ends up coding for a tyrosine. Since the stop codon is gone, translation continues until a new stop codon is reached further downstream. The result is a mutated, elongated protein.